ruined it. All right, a few pieces of housekeeping before we get started. So if you can, please make sure that you silence your phones. Photography is permitted, as long as, do you have any objections? Or, perfect? Yeah, get the good side. My wife says that's the back side. So social media policy, make sure you post everything to hashtag ABRF 2025. We've got the meeting map, our meeting app, and this will all be recorded, and the slides will be available afterwards as well. So, great. I believe, oops, yeah, this is the clicker that works.
Oh, it is.
I had this other one here.
I love it. Okay, well, thanks, guys. This is a little bit of a crossover sponsor talk. My company, we work in the omics space, and we're speaking with some experts in flow and imaging. But hopefully some of the features of our technology for single cell might be of potential interest in the broader customer and user universe that you guys work within. So we have a new technology platform for single cell of the sort with a sequencing readout. But something that may be of interest to the folks in this room is the simplicity and low cost of the platform. You guys might be familiar with single cell historically being a pretty expensive sample analysis modality, relatively complex one in terms of the workflows or the instrumentation required. And that's what you can see here on the left. Basically all single cell genomics technologies other than ours are powered by something that you would call partitioning, meaning the way you fundamentally perform the technology is by taking your sample of cells, putting each one into a different partition where you perform some sort of biochemistry on that cell, and that's what you ultimately need to do to achieve the single cell indexing and the single cell sequencing readout at the end of the day. Our technology, illustrated in sort of schematic here on the right, is powered by something that we call kinetic confinement. Now, I'll sort of skip a bunch of technical details, but give a concise conceptual overview. For any sort of single cell technology, be it partitioning based or solution phase, the fundamental challenge is to take your sample of cells, label the nucleic acids of cell 1 with index sequence 1, label the nucleic acids of cell 2 with index sequence 2, so on and so forth across all of the various cells in a sample, so that when you then put those nucleic acids on a sequencer, you can informatically deconvolve all of the sequences that came from cell 1 because they have index 1 appended to them, those from cell 2 with index 2, et cetera. Now, existing technologies work. They work analytically. They work in terms of the insights you can drive, but they're quite complicated. They're generally quite expensive. Our technology on the right, instead of taking your cells and partitioning them for the indexing reaction, we basically do the opposite, where we've engineered a new type of indexing reagent, which is a bead, that carries index sequences directly to cells. So rather than needing to take your sample of cells and partitioning them with microfluidics or other complex workflows, in a single PCR tube, you add however many cells you have in a given sample, alongside a corresponding number of these indexing beads. The beads physically pair in a one-to-one manner to cells, driven by what we call cell binding moieties on their outer shell. Then in that same PCR tube, you lyse the cells, you index them at the single cell level, and then you proceed with a very simple library prep reaction before loading on the sequencer. So whereas most single cell technologies involve quite complex equipment or workflows or both, if you have a PCR tube and a PCR machine, you can perform our technology. And this is something that we think might be of interest to folks outside of the traditional genomics domain because the workflows are simple, there's no CapEx outlay, and actually many of the intermediate steps that you get from a typical like FACS reaction, for example, have roughly the right cell numbers that you could use as the input to our assay. And so hopefully some folks in the audience may at least have their curiosity piqued a little bit to learn a little bit more over time. I'll skip the details of this slide, but this is a common benchmarking experiment for single cell, something called a species mixing experiment. Each dot in each of these 4 samples is what the assay thinks is a single cell. The input sample that we've given here to the assay is a 50-50 mix of mouse and human cells. The vertical axis is the number of human genes in each cell. The horizontal axis is the number of mouse genes detected in each cell. And what you should see if you have a high fidelity single cell reaction is basically all of the cells have either only human genes or only mouse genes, and very few cells should appear to have some mix of both human and mouse genes, because that's not what the cells were like at the original. And that's basically an artifact that you see in any single cell technologies. There's 4 different samples here, basically with doing the indexing reaction in normal PBS buffer versus doing the indexing reaction in increasing concentrations of our special buffer that provides this kinetic confinement. And you see on the right, when you have full kinetic confinement, you get a very large number of cells, you get high molecular sensitivity in each cell, and you get low doublets and low noise. And this is just an analytic demonstration that the single cell reaction and the single cell chemistry we've developed is working with high fidelity. For those of interest, we came out with a bioRxiv paper last quarter when we launched our technology for the first time commercially. And this paper, which you can access, has got quite a lot of technical detail about the technology, how it works, different example data sets, et cetera. So this is what the technology looks like. It's an entirely kit-based, instrument-free platform, and it's just a couple of boxes, and all you need is what you get in these boxes, and then basically a PCR machine. So it's very flexible, and it's very straightforward for people to pick up and implement, even those without material genomics experience otherwise. And we have 8 and 16 sample kits that are on sale right now, and we'll be coming out with a 96 sample kit later Q2. Something that's important to note about these is if you've done any sort of molecular or cellular biology, you will probably have the technical skill to be able to perform this kind of single cell. It requires no specialized expertise or equipment. And another thing I'll say is that it comes in 8 and 16 sample and soon to be 96 sample kits, but you are not constrained in terms of doing all of those samples at once. So like if you buy a 16 sample kit, you could do a couple samples today, you could do a couple samples next week. If you have a user that wants to try just a single sample this week, they can do it, and then they can go back for more the next week or the next month or whatever. And that total independence in terms of sample size and experiment number is something that's pretty unique to our platform within the single cell universe. It's a single-day workflow to go from cells to sequencer loadable libraries, and it's basically that upfront bead pairing and then indexing reaction, and then very standard like RT-PCR library prep. So if you've done like RT-PCR, RNA-seq, or similar library prep workflows like that, this would be very familiar and tractable to you. This might not be of strong interest to the room here, but a lot of our customers are interested in doing relatively large-scale experiments in the sort of plate scale, and the simplicity of the workflow and the cost profile of the workflow ends up making it quite attractive for that. The last quick thing I'll point out on this slide, after you pair those indexing beads to your cells, you can just freeze the samples. In that buffer, there's a cryopreservative reagent that we've added in, and so you don't have to do this entire workflow at once. You can do the first hour and a half of it, flash freeze on dry ice, freeze in -80, and collect those samples hours later, days later, weeks, or actually up to several months later if you want to complete the rest of the workflow, and then load the samples on the sequencer. So it sounds like a minor technical detail, but actually that's a sort of convenience factor that a lot of our customers have found quite attractive. Sure.
How many cells per sample?
We recommend you start out with anywhere between like 2,000 and 8,000 per sample. And then you can get, depending upon that number, you can get somewhere between 2,000 and 5,000 out at the end of it that have been indexed and sequenced. Sure.
What kind of receptor do I need on the cell surface to bind?
There's 2 modes to the chemistry. One is cell agnostic, where actually anything with a plasma membrane will bind to the beads, and that's a universal version of the chemistry, so you don't have to have any prior knowledge about the protein content of the cell surface. That's how most people use our tech, but one thing I'll quickly flip to. I have way too many slides for this. I have a couple minutes left. Ask a couple questions, and then we have a booth here, booth 126, so you can ask more questions. It's a great question because an alternative second version of our chemistry is cell-specific, meaning that you can basically coat our beads with an antibody of your choosing, and then use that to pair the specific cells of interest for single cell indexing and sequencing. Analogous sort of to like a MAX reaction, if you're familiar with MAX. And this is just a proof of concept experiment we did there where we started with PBMCs and used either a pan-PBMC antibody, I think CD45, or T-cell specific or a B-cell specific antibody. And you see the very strong enrichment if you have specific antibodies for the cell types of interest. So I'll quickly flip back through some data. Please do, if folks here are interested, please do stop by our booth. We have a couple of folks from our technical and our commercial team. Very happy to answer any questions. The technology works in a variety of cell and specimen types, normal cultured cells, disaggregated solid tissues, PBMCs, whole blood, stuff like that. It's quite scalable. You can do the assay in plates, and this is an experiment we did just to demonstrate the sample scalability, where we had a single scientist take several hundred samples from cells to sequencer loadable libraries in a single day, which for most other single-cell omics technologies would be a very complicated, very expensive, and much longer experiment. Quickly flip to this. We have a variety of early customers that have used our platform for a variety of applications with a variety of different cell and specimen types. It's pretty universal technology so far as we and our customers have seen so far. We've had some users validate against a variety of comparison measures, including FACS and flow, including other molecular readouts, including benchmarking our tech against other single-cell technologies, and they all look pretty comparable, actually. So I will pause there before time. Thanks for listening, and please do stop by our booth if any of you might be interested to hear a little bit more. Thank you. Thank you. Appreciate it. Thank you.
And now for something completely different. Perfect. So what's new in imaging cytometry? A whole lot has changed in the last 4 to 5 years, right? We've gone from 1 instrument to roughly 10 now on the market. So we wanted to give everybody here a full recap, all the technology, how it works, and then we have an open panel session with a few different core directors that have the technology and love it. So conflicts of interest, I have an Attune SitePix on the advisory board for the Attune SitePix and the NanoSelect Verlo. Bert Ladd from the University of Loyola has an image stream. Dave Adams from the University of Michigan, image stream and an S8. And Asia Rieger, who could not be here with us today, no conflicts of interest. So we'll go over some of the recent advances, the current instruments on the market, future speculations, and then open it up to a panel session. At the end of this talk, we will have a full instrument table, a full comparison on everything. Don't worry about this. We're going to have a QR code again at the end. You don't need to write anything down. This is also a 90-minute presentation that we've tailored down to 30 minutes, and that QR code will have the full 90-minute slides for you. So what is an imaging flow cytometer? At some point, we had to draw a line and say, this is what we're calling an imaging cytometer. So for the sake of this talk, it's going to be events flowing through a flow cell and that it has to generate an image or imaging data. The other one, before we can really dive into what's new, we really have to talk about where we've been. And largely everybody's perceptions on imaging cytometry has been shaped by the MNIS image stream Mach 2. And when this instrument came out, everybody was really, really excited. You had the microscopists that said, great, I can get my pretty image on a microscope, and now I'll get all my data on this thing. You had all the flow cytometers that said, great, I'm going to take my entire assay, port it over, and get a whole lot more information. And so core directors went out. They tried to get S10s. They begged their admins for money to get this thing. They got it. It got installed, and after a month or two, usage kind of really tailored off. And so for us to understand where we're going and how far we've come, I think it's important to address a lot of the misconceptions about the image stream and its utilization. And I'm not saying that I'm right in this, but I've tried to talk to as many people as possible to get a sense of why this instrument wasn't utilized and didn't seem to meet the expectations that everybody thought that it would. The first one is going to be speed, right? It was billed as having a max speed of 5,000 cells, and while that's certainly possible, most people are running it at 100 cells per second. And the reason for that is they're using hydrodynamic focusing, but you still get the cells that kind of snake around. And so you have to constantly adjust your focus to get those cells in focus. And so the samples are spiked with infrared beads, and there's this autofocus module that then tries to adjust the focus in real time to get all of your cells in focus. But then right off the bat, the people that said, I'm used to flow speeds, this is too slow for what I want to do. And for a lot of the microscopists, they said, well, I kind of already have a pipeline. This isn't providing that many more cells. So it wasn't really practical for them either. The second thing was you can get a lot of data from this instrument, but that's difficult. So when you're designing an assay or you're making a template for a user, it could take you 10 hours to say, here are the parameters that matter to get you the data that you need. But then that led to the last problem, which is oftentimes people were only using this to run 1 or 2 more assays after you got it set up. It didn't have the same return on investment that we think of with our cytometers, where I train a user and they're going to use it 10 hours a week, for the next 4 years. So I like to think of this a lot as the time I bought my grandparents a Nintendo Wii. It's a great gaming system. They didn't use it. That's not necessarily the fault of the Nintendo Wii, but possibly my perception of how they were going to use that technology. But again, understanding these misconceptions about imaging cytometry, do these things still exist? Are these still limitations? So the next big jump forward was with the Attune NXT, and this has been around since 2015, but fundamentally it works a little bit different. So instead of the hydrodynamic focusing that we have on regular cytometers, they have acoustic focusing. So it's producing a sound wave that is going to bounce all those cells into a single file line before they ever start mixing with your sheath fluid. And so during COVID times, Thermo said, I wonder if we can put a camera in there. I wonder if that stream is so darn stable if we could do that. So this has been used to allow you to run up to 1,000 microliters per minute so you can go really, really fast without it impacting your data quality at all. And when they put that camera in there, they found out that, yes, they could. So it's no longer a theoretical max of 6,000. You're just getting 6,000 cells. And this is a fully functional cytometer. So not necessarily an imaging cytometer as much as it's a flow cytometer where you're getting images automatically. There's no sacrifice. They started with this 20x objective, and it was only bright field. But again, I can see these are 2-micron bacteria with amazing resolution for a 20x objective. And it had some basic imaging parameters. But this really got the ball rolling because, again, I'm already using the instrument. Why not get the images as well? And that allowed people to go, this kind of has some utility. Next big jump forward was in 2023 with the FACSDiscover S8. So now we're getting imaging 10,000 images per second. We're at flow cytometry sorting rates and speeds. And the really cool thing here is they don't have the acoustic focusing, but they found a different way to get at that speed and without using a camera.
The blue laser beam is directed through an acousto-optic deflector and split into 100 separate beams. The unique optics cause each beam to blink at its own frequency. As a cell passes through the array of beams, each beam generates a signal that is collected onto a photomultiplier tube. Frequencies are mathematically distinguishable, making it possible to separate each beam from the complex combined signal generated by a passing cell. The frequencies are constructed to produce a complete image with VD Cell View image technology.
I mean, isn't that crazy? Like that we're making images without a camera? I mean, this is the part that I love, is just the new things that people are coming up with constantly. But here we are, 2025. 4 years, we went from 1 imaging cytometer to the market, but now there's 10. There's got to be something to this, right? So let's talk a little bit more about all the things that now go into the workflow. We're used to this diagram for lasers, right? It makes sense to us that I get a side scatter, that I get forward scatter. But if I took that detector out and put a camera there, that's going to be a dark field image. Likewise, if I remove my forward scatter detector, that's going to be a phase contrast image. And if I remove that obscuration bar, that's where I'm going to get my bright field image. So often I hear, what am I going to do with a bright field image, right? Like I need fluorescence. But think about what you can already tell with just your forward and side scatter on your instrument. And that's just a single data point. So now what can you get, again, with all the complexity going on between cells, with just the way that light is being bent through it? There's a whole lot of information there. When we do analysis, we can break down our imaging parameters into a couple of different categories. Shape, right? We're deciding how eccentric it is, what the major minor axes are. We have intensity parameters, the distribution of intensity across the cell. Entropy being, how speckled is it? Do we have more dark pixels, more light pixels? And now that we do have fluorescent images, we have fluorescent parameters. So colocalization, how far away is the fluorophore from the center of the cell? How spread out is the fluorophore? Once we have all these parameters, it's important to understand how are we getting those sort of on our cell. And that's what an image mask is. So I have my cell, and then all the softwares are going to make a mask or draw an outline around that cell. This is then what all the analysis is done on, is on that mask. This is important because you can have bad masks, you can have good masks, you can have perfect masks. If the only thing you're trying to figure out is this a cell or is it debris, you're going to be fine with a good mask. If you want to start doing label-free phenotyping, if you want to look at like membrane proteins, you really need to make sure that you have a perfect mask and a software that's going to allow you to really kind of iterate on that mask to make it perfect. So now we've got imaging parameters, we've got a whole bunch of them. Is this still a really complicated workflow? And the great thing is that it's not, because computers have gotten way faster and the analysis has gotten a lot easier. And what we're basically using is high-dimensional reduction. This is the really quick overview. If I gave us all these shapes and we had to sort of break them into different populations, there's the way the cytometrist is going to do it. I'm going to start making my scatter plots. I'm going to start breaking things out. And then there's the way that my 6-year-old son is going to do it. He's just going to sit there on the table and group them all together. In essence, that is dimensional reduction. We're taking hundreds of parameters, we're grouping cells together, and putting them down into a 2D plot. How is this helpful? Because I can take a treated and untreated sample, combine that data together, do the dimensional reduction, clustering as well can be done pretty much automatically at this point, and then I can separate those files back out. Really quick and easy then to say, well, what was the difference between my treated and my untreated cells? Less rectangles, more triangles. The only question is, well, what's a rectangle and what's a triangle? But again, the workflow is so much easier to work through now. So a practical workflow for me running samples on one of these instruments, again, 30,000 images, takes me 5 seconds. I do all my processing and feature extraction. It took me 99 seconds while I was in a Zoom call and I had 50 tabs open in Chrome. You know, the dimensional reduction and clustering, again, 10 seconds. And I end up with this. And so the first thing I like to say to my users is, how many clusters were you expecting in your sample? And for this case, I said they're just HEC-293 cells. There's just one cluster in there. There shouldn't be anything more. And when we add the clustering, right, it predicted that there were 5 different clusters. And the debris makes sense, the cells make sense, but the fact that the software can so easily pick out that there are 4 different populations within just that cell type, it's kind of amazing. And you may look at these and go, well, they're not really that different. Well, I guess they are. I guess these are not as round, and these have some more white dots in there. But that's the thing. There's so much information just in a bright field image, and we can add even more on top of that. It gets more exciting when, again, you concatenate your files. Let's mix them together. So I combine this untreated and a treated file together. And for this sample, it's just a CRISPR screen. They knocked out a gene in the HEC-293 cells. And now I label the treated and the untreated, red and green, and there's a difference. How's there going to be a difference in morphology when all I'm doing is a CRISPR screen? But that's the exciting thing, is it's rare that I don't find a sample that I can't tease apart just based on morphological parameters. And this is exciting because you show this to the users and all of a sudden they're like, okay, this does matter. Now I got to figure out why. And that's the beautiful part because now that's on them, right? No, we still like to help. But it's incredibly powerful. If you really want to see the full stretch of this, I got a great talk where we were able to actually map morphological parameters onto autofluorescence, and then you're able to sort those out on any sort of traditional cytometer. But again, seeing what is actually possible with just images without inhibiting your workflow is amazing. And that's the question to you. What could you do with the ability to take images at the same rate that you're currently running your cytometers right now, to get hundreds of more parameters, to have a simplified workflow and be able to sort on these images? And that's really where you're going to come up some amazing ideas. So the current instruments that we have out here on the market, I reached out to all the different companies. I asked them this basic series of questions. I tried to reach out to core facilities that own these different instruments, users that had them in their individual labs. I got general costs that I'm going to be able to share with you today. These are list costs. We all understand that that is not the price that we all pay, but that is just going to allow you to kind of bin the different instruments. Same thing when I tried to calculate the actual cost of consumables and talking to cores, its usage. And then I gathered 30 instrument-specific questions from cores around the world. And I said, when you want to buy an imaging cytometer, what are the stats that are important to you? So we're going to go through the transmitted light ones first, then the fluorescent ones. And we're going to start off with the Yokogawa flow cam.
After a liquid sample is introduced into the top of the flow cam, a software-controlled syringe pump pulls it through the flow cell and past the flow cam's optical system. Using a stroboscopic flash to eliminate motion blur, a camera captures images of algae and other particles in the sample through an objective lens. Simultaneously, an optional onboard laser excites fluorescing pigments present in specific organisms, enabling the flow cam to automatically distinguish between groups such as cyanobacteria, diatoms, and other types of algae. In real time, the FlowCam software visual spreadsheet automatically separates and catalogs the images of each organism. Using built-in image recognition tools, users can classify organisms, estimate biovolume, count cells, and calculate the abundance of each taxonomic group.
And so roughly this unit comes with 4 different objectives. They're paired to the flow cell and the syringe pump, and they're just swappable. You swap them out for what you want to use. But the field that's really been driving a lot of things forward in imaging cytometry has always been marine biology. This is sort of their go-to instrument. It doesn't have hydrodynamic focusing, so some of your images may or may not be out of focus. It's a continuous feed system. There's a reservoir on top that they're just loading the samples in, and it's going to sit there and run. When they're out on a boat, they've got a pump that just kind of constantly is in there, constantly measuring and exporting the data. The interesting thing here is they have a whole bunch of different models. They can go really small. We're going to talk about some of the large particle instruments later on. And again, they have a macro model that can do the same thing. Because there's no sheath fluid, the consumable costs are just the QC beads, which last forever. It has a fluorescent module. It has 2 detectors. It's nowhere near the sensitivity that we're used to. It's practically just, does this contain chlorophyll or not, as sort of their binning populations. So next, Thermo Fisher Attuned Cypix. Again, this is the one with the acoustic focusing that we just talked about. What I'm going through here is the software, and this is kind of the same for almost all the different imaging cytometers. You can take a gate, you can move it around, immediately see the images that are there. You can do this in real time or after acquisition. You can click on an event and say, show me where this is on the plot. Some of the really cool new things in their software is a similarity finder. All you have to do is click on an individual cell, say, find me everything that's similar, and in 2 to 3 seconds, it does the calculations. And then you can identify all of these incredibly rare populations, which this is, think about like 0.1% of the population. And if you go to the bottom, you find all of your rare events, the things that are the most dissimilar to it. Dimensional reduction, again, is included in the software. I'm going to go ahead and pick the stats out here that are the most intensive, so this should take the longest to run. Well, that's going through. The other one is you can change your parameters on your X and Y axis to look at all the morphological parameters at any time. You don't even have to save images in a lot of these softwares once the analysis is done. It kicks off to a second program, so it doesn't, again, slow down your workflow at all, and that was 5 seconds. This is how long it took to do the dimensional reduction. When that's done, it loads the file right back in. And so again, you don't even have to keep these files in terms of what you're going to export for your users. And there it is. I've got my full dimensional reduction, and you can immediately start clicking around with your users and say, what is this population? What's that population? Is this what you were expecting?
Sorry to interrupt, it's a really great flow here. I'm a total newbie to this, so are you just talking about standard just imaging, or are we doing anything with fluorescence?
Yeah, well, this is bright field images, but the cytometer itself is a regular cytometer with fluorescence.
But are you looking at any fluorescence images, or just standard imaging?
This is just standard bright field at this point. Yeah, great question. But again, it's just that easy. The flashing is because I was recording this on a remote desktop. Our instrument gets used constantly, and the only time that I'm able to make any of these kind of videos or screen recordings is, at night. So incredibly easy. The other one that's interesting is that the fluidics is so darn stable with the images. This is what it looks like when you're collecting in real time. I actually use that as a QC metric instead of like my CVs, because I will see that these start drifting, and kind of making that sort of snake pattern or not perfectly center far before I will ever see a difference within my CVs. I think that's pretty cool. Image rates, it goes up to 6,000 or a minimum of 3,000. It depends on the size of the image that you want to collect. But because this thing can go so darn fast and it's one of the fastest autosamplers on the market, it really has a great use in high throughput sampling with morphological parameters. You don't have to add any luminescent genes and stuff like that in.
One more question.
Please.
Once you've clustered, once you've identified images, can you backdate to the fluorescence data?
Yes. And so for the consumables, it's a little more costly because of the Thermo Fisher sheath fluid that you have to buy. So sorters now, those were just analyzers, just bright field or transmitted light images. We've got the deep cell REMI. And so here's where you load in your cleaning solutions, touchscreen to look at what you want to get. You're going to load in your sample as like a 5 mil fax tube, and then all the sorting is done on one of these little microfluidics chips. Your sample comes through, it's going to collect the images, and it's either going to put it over to trash, or it's going to put it into one of these wells here. The kicker is that these wells can only hold 3,000 cells each. Their model is, again, just morphology, but they've got a really good objective. It's basically making a UMAP, and then you're saying, these are the clusters that I want to sort. You can also train it by running through macrophages, different lymphocytes, different cell types, so that when you run a mixed population, it can find them again. The other big one is they have 115 imaging parameters, 51 morphological, and they have these AI-derived parameters from their human model where they basically looked at millions upon millions of cells, and they've asked AI to say, can you find new ways to differentiate these different populations? Probably know the least about this instrument. I reached out to users, I reached out to their FASs, I reached out to the company. Nobody was willing to provide any information, so take that with what you will. But again, the cost for one of those chips as a one-time use chip is roughly $250 to sort 3,000 cells per well. Next iteration of this kind of technology is the ThinkSight Vision Sort, and they got a great video that explains that.
Vision Sort is a multi-laser dual-mode flow cytometer, powered by Ghost Cytometry. A new label-free cellular fingerprinting technology that operates on top of the standard fluorescent-based cell analysis and sorting found in traditional cytometers. Unlike single-point spot lasers, Ghost Cytometry uses a structured illumination to measure scattered, diffracted, and absorbed light from individual cells. For each cell, a collection of data-rich profiles is generated, encoding morphological features like shape, subcellular structures, extracellular detail, and the spatial distribution of fluorescent signals. Together, they generate a unique cellular fingerprint for each cell, representing its multidimensional morphological profile.
And so you can think of these sort of like what the S8 was doing, but they're not going to put it back into an image. They're just going to do all the processing there. So there's not imaging parameters per se, but they're still getting all that information and that data back out. So this system actually does contain some lasers and detectors. And what you're going to do is either do a supervised or an unsupervised training. So I could start here, add a die, and say that in the future, I want to do label-free sorting of the low-expressing population and the high-expressing population. That's the supervised training. You tell it, these are the two populations that I want to sort out based on my fluorescence later on. It then calculates its confidence that it can separate those two populations out, label 3 the next time that you run things. You can do that also in an unsupervised manner. You just load your sample on, it'll give you the UMAP, and then you pick. I think these are the different clusters that I end up wanting to sort out. Other interesting thing here is that their dark field is both a forward and a side scatter, so they're getting that information from two orthogonal angles, which is kind of pretty similar to holography, if you're familiar with that imaging technique. The other one is they just announced at SLAS that they're coming out with an analyzer. This instrument, being microfluidics, is a little bit slow. It's a little bit costly because you have to have the chip each time. But their analyzer is going back to a flow cell, and it'll have an integrated 96 well plate, so you'll be able to do this same stuff, but really fast. Next instrument, the Union Biometric Copus Vision. This is a large particle sorter. It works very similar to an electrostatic sorter, but the droplets are so big, they actually use a jet of air, and that's constantly pushing the droplets out of the way. When you want to sort something, the airstream turns off, and it falls into your well plate for you. It's a collinear system, similar to like the Sony MA900 that we're used to, and it has a bright field camera, but you can't do any imaging decisions off of that camera. So it has 4 different models. Unlike the Yokogawa system, these are not interchangeable. You have to buy a whole new system if you want one of these different models. The other one is the profiling feature, which is pretty interesting. We're used to just gating on a certain fluorescence. Here you can actually gate on the number of peaks in a fluorescent detector, descriptive parameters around those peaks, how wide they are, how spaced out they are. And then you still get a picture of everything that it does sort, which is pretty interesting. Hard part here is, again, the cost of the instrument, $750,000. If you want to do everything large particle, you probably have to buy the 4 different models. And I will point out here that this cost was not provided by the company, but rather from talking to different users. So those are all the ones that just use transmitted light for all of their imaging. Now we're moving on to those that have fluorescent imaging. To keep with the theme of the large particles, it was always just the Copus Vision for doing analysis. There's now actually a large particle analyzer on the market that is fluorescent imaging based, the Bennu BioVelicite. What's really cool here is that they're using the acoustic focusing again. Instead of going into just a single wave, they produce 4 standing waves. So you can have a really chunky sample come in, splits it into 4 different streams that are all going to be perfectly, again, in focus because of the acoustic focusing. Then they can get the images. They do 3 different fluorescent images and then a dark scatter. The other one that's really cool is that everything that it runs, you get right back out at the exact same concentration. Run your sample, you pull it right back off. So if you're doing something like spheroids, you can just keep analyzing them at multiple time points. So their acquisition software is, again, like basic fluorescence that we're used to. To get the images and do all the image analysis, that's going to be after acquisition in a secondary software. And again, this is what things look like. What's cool is that although the imaging mask is 350 sort of microns wide, the acoustic focusing will always line up sort of modular things like these C ligands. So they're 700 microliters long, but again, they're all lined up, so you're going to get that perfect image in there. And this camera is actually sort of binning those 4 streams. It's one picture, and the software cuts them up into 4. So it's also possible to go much larger than the 350 micron. You just have to sort of bin the images back together again. Now we're back to the Cytec Amnis image strain, the Mach 2. So this has 12 different sort of fluorescent channels. Usually it's 10 with 2 different bright field channels. You have to buy 2 different cameras to get the full sort of spectrum. It can do dark field images as well as the bright field images that you can collect in any channel. The way that this one works is sort of this time delay integrated CCD camera. As it's going through, it's hard to see the individual like pixels in there, but it's passing the charge from one onto the other. It's like increasing your exposure time on a microscope, and that's how they're able to collect way more information and data, and they can get really, really sensitive measurements. Again, this is going to be their software, and as you can see, that was sort of that snake effect that I talk about. They're not always in a perfectly straight and centered format as they go through the flow cell. And so for those that don't use acoustic focusing, we're generally using a statistic here called the gradient RMS. That's sort of your measurement of focus. And so even if you're collecting things at 100 images per second, again, maybe only 50% of them are going to be in focus and usable. But the software itself is really easy to use. You can switch between the different objectives, the different flow rates, turn the different lasers on and off really simply. Overall, with the power of this instrument, you can do anything on this instrument if you have the time. 86 imaging parameters and 22 functional masks. What that means is that I could have one mask on that orange cell, another mask on that green cell, another mask on the points where they overlap, and I get 86 parameters for each one of those. It's truly an overwhelming amount of information. Because of the TDI CCD camera, they can actually detect things down to 27 nanometers, so it's phenomenal for small particle work. And again, it's an unsurpassed resolution in terms of its fluorescent sensitivity. The thing I'll point out here is the consumable cost. Those speed beads, those infrared beads that it uses for the focusing are free if you have a service contract. If you don't, that's roughly how much it increases the cost of your consumables. They have machine learning and AI-based software platforms that make the analysis incredibly easy, but you're going to pay for those as secondary softwares. And then again, these are their stated maximum rates, and what I've put here in parentheses is what the user-reported maximum rates are for those different objectives. Now we're on to sorting. We already talked about the S8, right? That we're using the acousto-optic filter to make roughly histograms that we're putting back together as an image. For the sake of time, I've combined the S8 and the A8 because they work very similarly. They have very similar stats. I'll point out where they don't. It's just that you can sort with the S8. This is what their software looks like. And again, we can see that you can just click on any cell and see the image. Because how many times have you been out there sorting and you're like, I swear they don't have a single cell in this sample. And it's now you can show them that that's the case. So that's pretty powerful. But it's just great for cleaning up and refining any of your gates with all of this imaging technology. It's a lot like when Spectral first came out, and people were like, why do I care about Spectral? Like, why do I need more information to separate things out? I can already separate out FTSE and PE. Well, that's what you're adding. You're adding in sometimes hundreds of new parameters. It's just going to refine all of your gates and all of your populations that much better. And that probably means my battery died. Let's see. We lose AV? Thank you. Advance them from back there. Is it frozen?
Kind of what it feels like running the image stream.
No way a reflection of the BD software. Should we just close it and restart it? Yeah, we can ask some questions now.
The previous platform, the Cytec, people use that to look at like binding events between, say, nanomaterials and cells. Say that again? So they said, according to the stats there, it could resolve down to 27 nanometers. So that seems like that might be an application for looking at binding events between, let's say, like a targeted
So when we're doing, you know, EV work on the instruments, it's important to be fluorescence because of the original camera. You're not seeing much in white filters, but it's very fluorescent.
Right. That's okay. It's easy to sort of solve for an material, right? For whatever thing it might be. Just like put a probe on their backbone or whatever the material is made out of. So it seems like an interesting case. It sounds beautiful. I've had it before. Yeah. So, but that would be, so let's say, for instance, well, that's probably a pretty technical question, but you've got a material that has like a really high aspect ratio. Let's say it's much longer than that on its long dimension, but on the short dimension, you're looking at something with like, 2 to 10 nanometers. That's like the sort of materials I'm working with. Would you be able to resolve anything out of that, I wonder? I don't know. I don't know if I expect a specific answer to that.
You said down to 10 nanometers?
One of the classes of nanomaterials I work with are like these high aspect ratio materials. So you get something that's like up to a micron in length or even more than that, but they're maybe 5 to 20, well, 2 to 20 nanometers in diameter. So it's like a pivotal type material.
Isn't,
So it seems like you might be able to catch something that's the fluorophore based on there, just messing around with fluorescence density or something like that on the baseline.
it really depends on its scattering properties.
Yeah.
So like we're looking at a lot of sizes here, but that's also based on biological materials and their scattering properties. It's just like when you use like NTA for characterization, it says it can go down to 20 nanometers. That's only when you're using gold particles. But if it does scatter light differently, that's where you might be able to pick it out with just differences in your bright field and your dark field images, because that's going to scatter and then you can illuminate them all without any fluorescence. But yeah, we talk about that a little bit later too.
Short answer is send me a sample.
Yeah. So stats on the S8 and A8, again, 3 different fluorescent channels. The big kicker here is they don't have compensation on the images. So you can't tell, again, if it's this green leaking into my red or vice versa, but that is something coming out on a future software update. Imaging rate is actually faster on the A8. They go up to 12,000 images per second. Flow rate, when you're operating it for analysis, it can go a lot faster. When you're doing the imaging, there's a hard cap that this is how fast you can run for them to get the images. It's cool that you can see all the images right after you sort them when you're doing sort of the index mode. But again, just looking at a GFP sort like we normally do, it's just extra information, right? Is the GFP spread throughout? Is it localized? And then the caveat here for the cost, BD wanted me to tell you all that the S8 is $500,000. That is a mid-range unit, whereas I've been showing the fully loaded units for everything else. Last one, the NanoSelect Verlo. This is a microfluidics-based fluorescent imaging sorter. It's basically rastering the laser back and forth to produce the images. And again, microfluidics, so using sort of a PTZ device to push the cell over into a different channel for sorting. Cool here, again, they're gonna have 9 different fluorescent channels off the 488 and the 561 laser. It has compensation. They've got the bright field, the phase contrast, and the dark scatter as well. A little bit slower, again, because it's a microfluidic system. But if you're gonna do sorting and with label-free, again, the resolution is pretty darn impressive for what it is. Consumables, again, a little bit higher because it's a one-time use chip. Other one I wanted to gather was pictures on all these different instruments. It's hard to actually get a sense when looking at, magnification, numerical aperture. So I tried to get raw images from users, right, where I could, so that you could actually see what large things look like, what medium-sized things and small things looked like. Again, we'll have all these slides available. Same thing for the large particles across the 3 different systems. And again, try to put the sources from the users that provided these images. Future speculations. This is where the field is going. The company that is closest to commercialization, in my opinion, is going to be Zomp, but I can't tell you how many individual labs there are that are already working on 3D imaging cytometers, how many folks at Cyto for the innovators and the technology showcase are going to be talking about 3D imaging cytometers. So it's not even, is imaging cytometry worth it? Like we're already moving sort of to the next stages out there, which is really, really interesting. The last one I want to talk about is high content imaging. Flow cytometry has gotten images, they're getting better and faster, but so is microscopy, right? With the spinning disk system, they're now taking images 1,000 times faster than they were with a regular laser scanning confocal microscope. So these high content platforms, we have one, and I can take all of the users that I have in my basic cytometer, put them on this instrument, and all the things that we as flow cytometrists may have hated about microscopy don't exist anymore. You just put a plate down, it's in focus, and you just go through an image. I mean, it's amazing. So this user here, it's just 3 colors, they're imaging 60 wells, capturing 23,000 images in an hour and 14 minutes, which puts them at 363 cells per second. That's pretty much the same rate that some of those imaging cytometers that we just looked at. But they also export the data as an FCS file. I don't have to train the users on how to do any image analysis. Okay, all the same lasers, filter settings that we have in our cytometers, but it also includes full environmental controls for long-term live cell imaging. There's no drift anymore. And the users don't have to culture their cells and put them into suspension. They grab it out, they put it down on the instrument, handles a whole bunch of different stuff. And again, the cost of these instruments is right around the same point that we are at for cytometers. So if you're not aware of these instruments, your microscopy cores are. And the last thing, when we're comparing our actual billing rates as a core facility, for our tuned site picks is roughly $36 an hour is what we charge. For this instrument, it's $24 an hour and $8 an hour if they're going to be running it for more than 4 hours at a time. So it begs the question, do these instruments have a place in core facilities? So with that, we'll turn it over to the panel. Ask us whatever questions that you have. Again, I've tried to put all the stats here. This is just the top 15. That QR code is going to have all 30 instrument stats across every instrument as you're trying to decide what matters, what doesn't matter as you're considering the purchase of an imaging cytometer. And make that big and easy for everybody here. Now, I told you that that was the 90-minute presentation, so if you try to download that PowerPoint today, you're going to be sorely disappointed because it's about half a gigabyte. So go home when you go to download that. But with that, we wanted to open up. What questions do you have? How can I convince you to love imaging cytometry as much as I do? There's a lot of cool stuff to love. If you don't have any questions right off the bat, I can tell you we bought our tuned site picks with the USDA equipment grant to replace an LSR2 that had 30% use time. We projected 5% growth over the next 5 years. Within the first year, we were up to 80% utilization on it, and now we're already purchasing a second. It's a regular cytometer. People are getting the images. When you walk them through that 30-second use case, they go, wow, I don't really even need fluorophores anymore. I'm getting so much more information. It's amazing. The other thing is, Michigan State University, we don't have an immunology department. So all of our flow is sort of this novel space flow. And again, the things that we're able to do with these imaging instruments is, it just kind of feels like you're in the wild frontier. And it's exciting to see PIs actually get excited again about, oh, this is cool. What does this mean?
How many of those folks are taking advantage of the imaging thus far?
Almost all to some extent. And then you have a range from, are my cells dead? Or they look okay? To, full-on label free.
Yeah, UMISH, it's a very, if you build it, they will come sort of situation. So with our first SA, we originally bought it because for years people are doing their image stream analysis in our core. Can I sort this now? No. Yeah, kind of. So we had an initial user group to justify the purchase. But for everyone else, even though they had no interest in imaging whatsoever, we made it standard practice to really optimize the images. It's really easy to do right, really easy to do wrong on that particular platform. But they're coming back after these longitudinal studies where they grouped all their things. I wasn't really seeing the differences I expect, but when I included the image data in that, suddenly I'm seeing that separation, which is proven really useful.
Can you talk a little bit about some of the applications that people have drawn into who have had preconceived notions of what their data might look like, but have then said, oh, shoot, there's a lot more in these images. Can you talk a little bit about the applications where you found that to be the case?
I mean, it's kind of depressing one would be someone's done like a nerve digestion and swears they have cells in there. And it's just like, let me show you. You don't. But for one that's a little more optimistic, I mean, there's just a lot of cases where we find like with some metabolic stain we're doing, it's like, oh, we can see based on like, the contrast of the bright field, like all these H texture features. There's just a million little things we can dig into and find. Trying to think of one that comes to mind right now. Yeah,
So we got a few.
Sure.
We're thinking.
give me a second to think.
We had a person that got all the way to publication. Reviewer said, go back and, get some more data on cytometry. We put them on the site picks. Their drug was highly contaminated, and all the effects were because they had a contaminated drug that had bacteria growing in it. And wow, could you see that as soon as you put the pictures on there? But there was no difference really on forward and side scatter. So that was incredibly useful. We have groups doing microplastic characterization in water, because you can pick it up. They look very different than cell types. For a lot of people, nobody says, I'm really interested in imaging. What does this do? It's always in the training. When we have a control sample, and you go, can I show you something for a few seconds? And that's just where the lights go on, right? It's getting users to use viability dyes and everything else. You have to show them sort of the utility. It's now just never been easier to show that utility. And then you show them that, hey, this is what people are publishing in Nature, and they go, okay, well, yeah, now I have to do it, right?
And even just, I'm thinking of a couple applications, showing people, convincing them that they don't actually have fluorescence if they're trying to do some transfection that's not working. It's like, you can just see that this is autofluorescence from here. I think it is really a seeing is believing, kind of what Daniel says. It's just like, you've been doing this experiment for a while, I'll pop on the image stream this day and just see what else we can dig out of here. It's just very flexible in that case since it's the same prep.
We've got you.
I was very intrigued by your comment that, again, you're showing the microscope and the postdoc that you see in Jay saying we really need, as they want to, microscope. I was then thinking back at some of the technology that you showed, like the fingerprinting technology, some of the scattering properties that you get. So I'm curious if, do you see like a family of problems that would be unsolvable or, or let's say a better fit for those cytometers that microscopy have a hard time with? Or is it gap kind of closing?
The gap is closing, right? If you have an experienced microscopist and an experienced flow cytometrist, like, there are ways that we could get to the same questions. And that's what I tell a lot of people is, it just comes down to what instruments that you have access to. If I was really looking at the two different platforms that I had and breaking them down, it's the ease of use of the software for your users. They don't want to spend a lot of time. It's easy to get that simple FCS data for, 10-color assay off of the imaging. They don't really care about a lot of the morphological parameters, which you can do there. It's a lot harder to get that information in a really digestible format, whereas I feel like a lot of the imaging cytometry companies have made that workflow really, really easy. But if you're looking at spatial information, like, it's nice for us to have both available.
I think like in the open source world, if you think of software that's missing, it's probably this easy to use sorting software that would take a bunch of images and kind of break it down into each individual cell and let you do all the cases that you can do in a cytometry.
Yeah, I think they definitely could have that. It's, as we all know, as core facilities, we're not the major purchasers of these instruments. And for high content, when we got that instrument, they were surprised that any core facility would want this, because it's mainly in pharmaceutical industries where they're running the same assay nonstop, 24/7, 365. You don't need all the same things. And I think that for a lot of companies, it's a worth the time investment to make them much easier to use, simpler software, whereas at least for the cytometers, they're a little bit more used to these core facilities. But there's a lot of room for them to grow.
Thanks. Great presentation, Daniel. Curious about your comment for the future. So regarding 3D imaging for flow cytometry, I'm curious for the community, right? Would you expect a similar throughput for the cell, or actually it is okay that since you're getting 3D, right? So maybe the throughput can be slower. That's one. And the second would be, what are the resolution? I think that would really help the community in terms of 3D.
So the throughput for Zonk right now, they can do about 200 3D images per second. It's not stated on any of their website and stuff, but if they reduce that down to 2D, I believe they can do 100,000 images a second. So it really shows you how much they're oversampling to produce those 3D images. Things can always go faster, right? I don't think that they're using acoustic focusing, but it's you got to start somewhere. But I think that's what they're all working on, is that same thing, right? How do we get as fast as the cytometer with the imaging? Could you remind me your second question?
Resolution, 3D. What would be like a useful 3D resolution that
Yeah, I think that's what's hard when we look at it. I mean, right, they're showing different cell cycle, and you can certainly see a lot there within a THP1 cell. I know that for a lot of these companies, they are working on super resolution approaches that is commonplace within microscopy to see if that is a cheaper and more effective way to get higher resolution. I also know that the resolution that this instrument is capable of is not something that they have released to the public. So I'm certain that a lot of these images are kind of older and that they can do better. I know that's not specific, but that's kind of where it stands.
Thanks.
Great discussion, great panel. Thank you so much for putting it together. Curious if in your studies you saw any trajectories on publications using flow imaging work, and if you can speak to any trends, it's going up, down, steady. If you did see that data, any applications that stood out that were kind of exciting that you maybe hadn't thought of or seen before?
Yeah, you guys.
If you build it, they will come. I don't know about your guys' core, but if it can be done by flow, we're probably doing it by flow. So now we're adding that extra dimension of data. We don't have any one particular group that's like, oh, this is exciting. People are just not, we're collecting this information. People are now just getting it. We do a lot of surgery disorder, with the cancer group. So the imaging, cancer cells historically on cytometers, the scatter is always interesting to look at, sometimes hard to tease out. Adding imaging capabilities into that is providing us a little more information. And here, image stream, great, you got the time, it'll do it. We're more excited about actually being able to separate out things that we weren't able to see before. And so we do a lot of, the tumor folk are doing a lot with that. We do a lot of worms because BD's focusing is really nice that you can take, take the worms and they'll line up nicely and not clog your machine. They'll actually go through young, which makes for some interesting images, that sort of thing. But yeah, we're pretty application agnostic in our core. We've got 300 different labs currently using the core, so we're doing a lot of stuff.
Application agnostics, great term. Yeah, I like to have a reputation of just we'll try anything once. But some interesting stuff we've done lately. We do a lot of stuff more in the immunology context, a lot of like immune synapse stuff, localization of things to the membrane or the nucleus. And then a fun one recently was looking at cells infected with a coronavirus and looking at we could actually see gastromin forms or pores forming on the surface of the cell because we could tag it, just see a little mouth on there. But for us, it's really about the localization and internalization question that we just can't answer by conventional flow.
Yeah, a little bit back to the kind of nanomaterial talk, but so what case uses have you seen for people sorting non-cellular materials in that way? So I know you mentioned microplastics and EVs, so is there any other random case use that you've seen in your course?
For the itty bitties, the teeny weenies?
Yeah, well, I was thinking maybe separating out aggregates versus really tiny stuff. That stuff that maybe necessarily you can't see with it, but it seems that maybe you could do that. I don't know.
Yeah, we've had groups do nanoparticles. We've had groups that have done little silica fragments. At least trying to get a sense of the characterization of how different those are. Things that people were doing in TM and AFM. As long as it's within the technical size range, it's so much faster than this on cytometry. We try not to tell our users this is how other people are using it, especially when we go to different departments, but just we have an instrument that can take images down to this resolution really fast, and you're automatically going to get all of this data from it. Is that useful? And that's where we've gotten all of these really cool and weird projects, right? People want to run a soil sample. That's fine. I can find the bacteria in there. We can find all the little bits of debris, and dirt and everything. It's always something new.
We do a lot of yeast too.
Yeah.
Can you comment on top-of-mind data management challenges and solutions that maybe that's
It's a whole other panel. But.
Indeed. Our solution was we don't handle your data anymore. Yeah. We throw Globus up and throw it in. Historically, we've archived for 10 years back in the old days, and then the data started getting bigger, and it's like, okay, we're only gonna do 5 years, we're only gonna do 3 years. Now we're just getting out of the business entirely, because the university's actual data storing platforms are best practices where we have a shared drive. It doesn't make sense logistically, but we are buying, as we're making that shift, we're buying 48 terabyte drives and filling them up every 2 weeks. It's just an order of magnitude more data introducing the imaging characteristics.
Yeah, archival Blu-rays that get really large, just whatever you can do to fit it. I think really it is a change in approach of, we'll hold on to your data long enough for you to download it, but then I can't be the steward of it forever. And I probably get a little too far in archiving data, but that's my approach for sure.
Do you have any suggestions on data management companies to invest in?
We'll talk.
Yeah.
So your first or second slide, you showed phenotypic differences with CRISPR perturbation, right? So is anybody using fluorescently labeled ATGC and getting a readout on this?
Not that I have.
No, I'm not familiar.
And the second question goes back to your first. So the Amnis machine sat fallow for a long time. So what, in your opinion, has brought back the revival? Is it applications? Is it functional genomics? Why the big revival now in imaging cytometry?
So in our core facility, we're split on campus between the north and the south campus building. The perfect instrument that could answer everybody's questions is a 2-minute walk away, and they will not use that. They will use the instrument that is right next to it. And I think it's the same thing. If they can do what they're doing and get more information, they'll probably use that information. If it requires them to move on to a new platform or do anything else new, more complicated, they won't. But really, I think it's that it was the SciPix and the S8 that you're not changing your workflow. Now you're just getting images. Like, why wouldn't you use that, you know?
So what was the first spectral cytometer on the market? That lovely little Sony machine, which it was a lovely machine, but it really didn't do much. It didn't really offer you a big spectral advantage. They sat on it, being used intermittently, time goes on, until Cytec pops on the scene and says, this is what spectral's actually good for. Cytec started selling a bunch of machines. Now we have the Sony ID7000s, we have Big Fee, we have the Xenium, we have the S8, we have the S6, but if you're running spectral on it, I apologize for, the life you live. The Amnis has not gone through any sort of revival. It's just, someone had a niche case and said, that's great. I can do this better without perturbing your workflow.
So we have an Amnis industry. We are in the boat. When you did that first slide, I felt all of this. One of the biggest limitations for me, and I don't know if this is different now, this is my question, was my users can get the SDS file and do their normal FlowJo. How do they get the imaging data? Is it the same where it used to be, where that's just done in the software where you run it? So it's really you doing it, and then they just still get the typical SDS file? Because I have the immunology users, like you mentioned, where they might want to gate on 12 different populations and then look at the images within that. Is that all done just on, the acquisition
Sure.
software? Or how do you then give that to your users to explore as opposed to you doing it?
I find that typically with a new project, I have to at least do the first round of analysis myself and show them, and I generally do that within the ideas software that comes with Amnis, or comes with the image stream. But there is now FCS Express can just take in the RIF files from the image stream, and you can do pretty much all the same stuff you'll do there. I'm not as familiar with FCS Express, so I can't speak a ton on that. But I, yeah.
Well, so that, not great with the image stream. FlowJo is really your only option when it comes to the S8. Can't imagine why that is. So you're doing some live image analysis because you're using it for sorting parameters. After the fact, you can extract the images from the CZW file, carry them over with your FCS file into FlowJo, and do the analysis that FlowJo offers. But in the end, each cell is being exported as a TIFF. So if you isolate, the populations of interest with your clustering or whatever, you can then take the TIFFs like any other image analysis software and, play Mary Hobb with them, all day long. You can get a lot of information. But the workflow, the after-the-fact analysis, the image stream is complicated because there's too much and it's slow. The concur to cell view to FlowJo, it's fine, it's just annoying and, it varies. But yeah, once you've exported the images, you don't have to come back. You acquired an Inspire, now I'm going to, analyze them in ideas. Once you've extracted those images and exported groupings of cells in the FlowJo workflow, you can play with them with any image software you want.
I do end up ping-ponging between FlowJo. The idea software is very thorough, but it can get a little clunky, especially on my poor little laptop. So I usually will find subsets of cells, subsets of features I'm interested in, take them into FlowJo where I can more quickly play around with them, and then come back and see if that's corresponding to something interesting on an imaging level.
And a lot of the other ones will have a processing software built in, so literally the time it takes you to clean, they now have an FCS file that has been appended with every new imaging parameter. It'll export as an ICE file or something similar that the users can handle. And then it's just showing them this simple workflow, right? Just do a dimensional reduction, and you can put that into a heat map or something, and I have a template that I give to my users that they just quickly pull up and go, oh, entropy is the way that these different clusters, differentiate. So it's become a lot easier, and the undergraduate students are way more familiar with this stuff in R and kind of pick it up a lot faster too.
So I come to this from microscopy, and I do have some questions about how they're defining resolution, because 27 nanometers, we're working really hard at building some complex systems to get to that. But I am curious, kind of based on that thought, how much you feel you need to understand the image formation, the optics resolution, camera details to really serve users and make good use of it, and also the analysis side.
That's a much bigger question. All these different companies are using different number of parameters for their images, and it begs the question, how many do you need to do the things that you're doing? Do you need 115? All the things that I have seen, even for the ones that do 115 parameters, it's not really useful to have more than 35, 36. You can do everything that you need with that. And now the copy's worn off. Could you repeat the rest of the question?
The theory of forming the image, how much does that need to be known to really successfully use one of these systems?
You can look at the images and see what you're getting out of there. I don't think that you need super high resolution, depending on what you're doing, but that's where we had the pictures of the images. You go that's probably going to be good enough for what I need. The users don't really care. They're rarely looking at that detail that you are for microscopy. Oftentimes, they don't really even save the images. They get the imaging parameters, they're happy, they know what those mean, and they just take their regular FCS file.
And, it depends whether you're driving research down to a platform or you're working with the platform and looking up at the research people want to do. There is, I think, more here than there are in microscopy cores, sort of a mechanics view of things. This is what I can do with these cells, these circumstances. You build a fairly encyclopedic knowledge of that sort of thing, but the fact that this uses this particular lens, it's generating images with this camera that runs this speed is not so important, other than when you say I can do better, and I bet I can replace this part with something else and go off book. So yeah, I would say it's interesting to know. The more you know, you're always going to do better work. But again, from a mechanics view, looking up, I just need to know what I can get, not necessarily how I'm getting it.
Yeah, it's like if you're really driving down at the absolute limit of what the technology can do, I think that's when it really becomes important. I'll admit the optoacoustic stuff is kind of wizardry to me still. Camera makes sense.
And to jump on to what she said, and you brought up a really great point, is that 100 plus parameters can get really cumbersome, especially if some of them are orthogonal.
can then say, well, these 6 clusters are really important. I want to sort on those. Am I going to see those 6 the next time I come through, or are they going to disappear and be another subset of clusters that really have no relevance to the last experiment? Yeah, so I don't know. I just really think there's a lot I need to learn more from my process.
Well, there's a reason when the image stream first came out, it had all these wizards to help you not look at those things. If you look at the BD, I mean, BD's initial primary limitation when they were designing was compute power. I'm really looking forward to the S9, which isn't a thing. I just have some ideas. But from that point of view, they've nailed down to these are the things that we think are going to be important to really distinguish something about your cell and separate it out. After the fact, as I said, you can look at so much more, but that's tailored to individual research. Image stream is not actually limited only to those, 115 total he gave, because, if you can describe it in words, you can get, you can create even more. It's not easy, it's painful to do. So it's a question of what's actually important. And I've always said, when you're buying an instrument, buy the most you can at that given time. I'm a firm believer in, when it comes to imaging, maybe less is more.
Yeah, very much agreed. I'm not in the habit of just popping every single possible derived parameter from an image stream and putting it in a UMAP and see what happens. Daniel mentioned the feature finder, the deep learning module they have. Those are pretty dang good. You pick true populations and at least gives you an idea of like, okay, I need to start looking at circularity, I need to look at entropy or whatever it is. And then, yeah, these derived features can get really, really complex. Like I'm looking for CD41 that's in a round shape but doesn't have little bulbs on it and there's no DNA. So like looking for platelets stuck to a cell versus like a megakaryocyte, for example. Like it gets really, really deep. And those can be all new features that you plot now.
I would say it's like, do you do spectral cytometry at all?
I don't yet, no, just the traditional, yeah.
For those of us that have, it's like anything else. You get a sense of autofluorescence is usually around here, that I can get you one mixing in 10 seconds, and then I can get you a much better mixing within an hour. You can always go deeper to really kind of reduce those error bars, but again, you can get something really quick that just answers the question, does this matter? And then you go a lot deeper. The other one that's really cool is BD has this plugin called Hyperfinder that a lot of other companies are working on, and it's the same thing. Just you get the clusters and then say, give me gating to get back there. And then you don't have to worry about the clusters anymore. You know that now I can look at 90% plus for circularity, and out that population, I can do this next, sort of parameter. And you get used to like, okay, here are the 5 that I end up using the most. And then the others kind of have more niche applications.
And good to be friends with your imaging people, if you have them.
And clustering is a wonderful tool for doing bad things. It's you have to put in the time because clustering will tell you anything. And like he said, Hyperfinder is great because you can acquire on your instrument, throw it out the FlowJo, do the clusters, bring the gates back in. It gives you those gates. Thermo, very similar on the Bigfoot, but you don't have to exit the software. You just say, give me a UMAP or a TISNI. The answer should never be TISNI. And say, this cluster is interesting to me. Show me the gates, and you can go live. So there's a lot more immediate clustering going on that's proving useful. But again, careful in what you do because clusters will lie to you as often as not. So.
Just following up on that point, let's say you take that and you reanalyze, you sort the cells based upon your cluster gate, reanalyze it. Does it ever fall into the same location on the cluster?
That's where imaging is actually, the additive of the imaging is actually quite useful because in a world where you have all the sample you'll need and we can go through and actually take a few moments to look at these clusters, you will find some cases where, hey, these are distinct clusters, but no, they're really not. In other cases, I didn't expect that, because just looking at fluorescence peaks, I don't see a difference here. But again, you're looking at the cells and it's like, cool, now let's go to sequencing and see what it is.
There's a slide in the longer presentation, this is a basic immunophenotyping panel, where we're used to that cluster, where you need to define your clusters, you throw in the imaging parameters, and every island pops out completely separate. And it's like, wow, that is much easier. But I know that that's our time. Hopefully this has got you thinking a little bit more about imaging cytometry. Feel free to reach out anytime if you have other questions. Thanks for coming.
And seriously, send me a sample and I'll tell you what you can do. I love playing with toys.
And I'll leave that up if you need the screenshot again.
Thanks, man. Thanks. Why not? Sure. Yeah.
Just with the site mix.
You staying in this hotel?
Like I've mixed 5 different subjects together so far. And it costs a lot just on my phone.
5:30.
I mean, it's just so cool.
Reception, lobby, registration desk.
Registration? Yeah, sure. Do that. Yeah. Go.
That's my workflow now. I run the sample on there first. I can almost always refine the do like you look at a GFP sample and like I want the top 1%. I can show them the top 1% is the apoptotic cells. You just aggregate GFP. You know, like it's always helps.
Similar enough to where you're like, okay, I can it really did need that part.